Okay, hi everybody. My name is Celia Chari. I'm here to introduce our second speaker for this new webinar series of the American Ceramics Society called Material Science, Pioneers in Art and Archaeology. We are so excited to introduce our speaker for the day, Dr. Madeline Meier. And before I go into the introduction and the bio for Madeline's talk, I would just like to mention that this webinar is hosted by the Art, Archaeology, and Conservation Science Division of the American Ceramic Society. It is one of 11 divisions of the Society, and this year we have a few things that we are organizing. In addition to this webinar, we're also hosting a symposium at MS&T. We are currently accepting abstracts for that symposium that will take place in Pittsburgh, Pennsylvania from the 4th to the 7th of October. Those abstracts are due on May 1st of this year, and we are particularly interested in student talks. So if you're a student who is working at the intersection of physical chemistry or material science and heritage science, we would like to encourage you to submit an abstract. So that's due May 1st. Another thing that we just want to mention, or I want to mention on behalf of the AACS division, is that we are currently able to cover membership for some graduate students that are interested in ACerS and learning more about our division and are not currently a member. So I will be putting a link on the chat that you can fill in with your information if you are interested in becoming a member and you're not already. This is only eligible for master's and PhD students, but I will also put the email that you send that form to. So just fill out your details if you're interested. There's no need to fill out the section on payment. You can leave that blank and you can send that to Vicki Evans, and as the division will cover you for the year. I think those are the only housekeeping notes that I'd like to share before the talk. So I think we can go ahead and introduce our speaker for the day. Dr. Meier holds a PhD in chemistry from Caltech, where she worked on fundamental light matter interactions of nanostructured semiconducting materials. Dr. Meier is currently a postdoctoral scholar at Northwestern University and the Art Institute of Chicago Center for Scientific Studies in the Arts, also known as USAXS. And there she uses scientific techniques, both non-invasive and microdestructive, to investigate cultural heritage works across a range of material classes. And today she will be talking about her work examining Featherworks, which I'm so excited to hear about, using USAXS Synchrotron technique. So without further ado, let's please welcome Dr. Madeline Meier.
So what I'm going to do is the classic Zoom check. So is everybody here able to see my screen as well as my laser pointer here? Okay, great. So thank you, Celia, so much for the invitation to present at the ACerS AACS webinar today. I'm really excited to share with you all, while not ceramics specifically, a lot of the techniques I'll be discussing I think would be really useful and interesting applied to ceramic materials. So examining a really unique class of objects, in my opinion, I think in the field at large of Featherworks, and specifically looking at the multimodal techniques we've been using throughout this investigation process, with a particular emphasis on some of the synchrotron methods we've been doing, specifically ultra small angle X-ray scattering. So an introduction to feather-based cultural heritage objects, or as I'll be referring to them, Featherworks. So Featherworks are a particularly interesting material, and because they're somewhat understudied within the field of cultural heritage. Additionally, similar to textiles or other more three-dimensional works, the geometry can be challenging to assess and during the analytical process. So that's really interesting in how we approach these objects and can better understand them. And so feathers present a unique opportunity to investigate coloring mechanisms in particular that I'll get a little bit more into. So I've highlighted two examples of these beautiful works here. One is at a Quetzal feather headdress, a Montezuma headdress made likely in the early 16th century using these really iridescent, beautiful Quetzal feathers. And the other is a Peruvian feather work of a miniature dress from what we believe to be the 12th or 13th century in the Metz collection. And so these kind of highlight just a bit of the range of the objects that can be crafted using Featherworks and really highlight the different techniques and the three-dimensionality of these objects. So what makes some of these objects so unique is, as I mentioned previously, is the variation in coloring mechanisms that can occur. So feathers are really unique in that they don't just have one coloring mechanism. The color can be pigmentary-based from the form of biopigments that are present, similar to our more traditional materials. And then as far as other coloring mechanisms, there can also be structural colorants. And so I've shown two examples here. Green color is particularly interesting because oftentimes it is a combined pigmentary and structural coloring mechanism that together produces the overall observed coloring. So for a peacock, kind of the classic example of structural color with its very iridescent wings, the green responsible for these feathering colorings are highly ordered melon granules or melanosomes. The rosy-faced lovebird has that combined structure that I mentioned, where a yellow biopigment and a blue structural color. So the birds themselves kind of do their own paint mixing in a way with the structural color and the biopigments. And then finally, greens are particularly interesting because there's only one solely pigmentary green present within feathers, and that belongs to the class of turacos. So the color is in fact named after them, and it's called Turaco verdin. And this is hypothesized to be a copper-based europorphyrin, but it has not been extensively studied or classified as of yet. So I'm hoping that someday we'll learn a little bit more about that. But that's not exactly what I'm focusing on today, but it's always an active area that I'm following up to see what the latest research is available on that pigment. So the objects I'll be discussing today are a set of calendar screen panels we've been investigating in collaboration with the Field Museum of Natural History here in Chicago. So for these set of panels, there's 12 individual panels. Each panel contains a religious symbol at the top, a flower based on the time of the year in the center, hence the calendar screen panel nature, and then another set of decorative displays at the bottom. The panels presumed to be created during the Qing Dynasty in the 19th century and was acquired by the Field Museum in an expedition in 1910. And although, especially in the blue regions, it appears as this is some sort of continuous medium or might be painted on the screens, in fact, when we look closer, we see the resulting areas, especially for the blue coloring, are there actually individual pieces of feather that have been very meticulously and precisely glued and adhered together to create these continuous works. And this particular technique is known as Chang Chui or dotting with kingfisher. So a little bit more about the Chang Chui craft. This technique involves gluing individual feather pieces to objects to create these very beautiful, elaborate and intricate designs. The most common feathers that are used are from the common kingfisher. They have this very bright, brilliant blue characteristic coloring, and that is very recognizable form of the craft. But the objects are not only made with feathers from kingfisher, of other species of kingfishers of different colors, as well as peacock duck and chicken feathers have also been found historically. The objects made using this technique range from anywhere from smaller hairpins and jewelry to more elaborate screen panels and headdresses, as shown previously. So one of the reasons we were really excited to examine these screen panels at the Field Museum, and they were of great interest to us, is the combined use of both pigmentary-based coloring, such as these painted insets in the bottom here, and what we hypothesized to be at least partially based structural coloring from the feather materials shown in these top two insets. So we wanted to see how using more well-established methods within the cultural heritage field, as well as introducing some new synchrotron methodologies, we could really fully capture these highly dynamic materials and also these mixed pigmentary and structural color systems. So a little bit more about structural color for those who might not be as familiar. So in contrast with dyes and pigments, structurally based coloring results from light matter interactions with nanoscale features. So these nanoscale features are generally on the same size scale or smaller than the wavelengths of visible light. So that results in really unique manipulations and interactions with visible light. A common example that's often used is the morpho butterfly. and that's picture shown here. It has this very bright iridescent blue resulting from the nanostructures depicted in the scanning electron micrograph that have alternating layers of keratin air. So the coloration mechanism for how these works is we have visible incident illumination that passes through because it's on the similar size scale. Only certain wavelengths are allowed to be transmitted. Others are reflected back to the observer and can constructively interfere, and they result in this very characteristic bright blue iridescent coloring. So not all types of structural color have the same morphology. So it's what also is very unique about structural color. Just like with a pigment, you can have many different pigments that result in the same overall color. You can have many different types of sizes and shapes of materials that result in different colors. So the morpho butterfly with their sort of nano Christmas tree or tree-like architecture is one example. But there's many other examples present in nature, such as our feathers, that we'll discuss a little bit further. What's also very interesting here is that because of how the coloration mechanism works, if we infill these layers with some other material instead of keratin and air, if it's still infilled with ethanol or sugar water here, we can alter the color. So because of this unique coloring mechanism, we can really alter these properties in unique ways that might not be possible with dyes and pigments. So these materials have fascinated artists and scientists pretty much since the beginning of time. And there has been a lot of work attempting to mimic these materials from nature or incorporate the materials directly into the work, as we saw with the Kingfisher panels. And in fact, one of the first man-made structural colors, to give a shout out to ceramics, of course, were luster-based ceramics from 9th century Iraq, where they had these very iridescent light qualities they made through their firing processes. So we want to be able to characterize the structural colorants found in archaeological and cultural works, but there are key features with structural colorants that are present that might create some challenges for us assessing these unique materials. And one of the most interesting parts about structural color is often that they have angular varying, or they change color depending on the lighting conditions and the angle that they're viewed. And so while that's really fascinating and that iridescence is what draws people to the structural colors, that can also create some challenges in measuring them and assessing their optical properties because they're not static in nature. So a common one of the best examples, I think, of this are the bird of paradise. So these two images on the left here are actually of the same feather sample. So when the feather is normal to the illumination, it has this sort of yellow or goldenish hue. However, upon tilting the sample, this changes to either a blue or green. So you really have almost the whole rainbow of colors from one individual sample. And this can be measured quantitatively as well. The graph inset shown here shows the reflectance as a function of wavelength. And for panel A, represented by the red line here, we see for normal illumination, we have a peak around 600 nanometers, and upon tilting, that shifts to around 560. So we not only see these qualitative changes in coloring, but we can assess them quantitatively as well. So that's important to keep in mind when we're analyzing these features. So not all structural colorants display such a strong angular dependence or range of colors. So for Kingfisher feathers will be the central of our work today. The feathers do remain blue even when tilted at large angles. However, the precise coloring of that blue changes as depicted here quantitatively, where they had normal incident illumination and the probe that was measuring the reflectance response was tilted at larger and larger angles. And so we found that while they still remain blue, the maximum reflection intensity goes to shorter wavelengths as the angle gets larger. Additionally, something important to note when examining feather coloring across the same species or even with individuals of the same species is there can be a decent amount of natural variation depending on maybe the bird or evolutionary characteristics of the subspecies. So we first started our investigation by verifying the nanostructure responsible for the coloring in the blue common kingfisher. So on the left is the image shown previously of the common kingfisher, and we turned to focused ion beam scanning electroscopy to aid in the visualization of these nanoscale features. So we specifically did FIB-SEM, or focused ion beam SEM, because of the benefits in biological sample prep and 3D visualization. So recent advances in plasma and FIB technology now have better access to work with biological samples. And so we can not only see the morphology from a more two-dimensional perspective, but you can take slices throughout the sample that really allows you to put together a 3D volume rendering. Additionally, we can also see the top and bottom layers there versus we had just peeled off that layer entirely, which lets us better understand the materiality as a whole. So this is inside one individual feather barbule here. And so because this is a little tough to visualize the structure here, we have a more detailed inset. And this really helps us visualize the actual nanostructured morphology that's responsible for this really brilliant characteristic blue coloring. So the structure responsible for the coloring in the common kingfisher is a semi-ordered array, what I like to call a nanosponge-like appearance. And the structures of the features are on the order of 200 nanometers. Historically, it was thought that the coloring in these kingfishers might have stemmed from a similar process to why the sky is blue. But with scanning electron microscopy and quantitatively assessing these features, it was in fact proven that it is a result of coherent scattering. So even though these appear somewhat disordered, if you look at the local scattered order, they are still very highly periodic and have that structure needed to produce the structural colorants. We then turn to hyperspectral imaging to investigate the optical properties of the feather details on the screen panels further. So one specific panel here, we have a highlighted inset region here, and the spectra on the graph correspond to regions of interest for the blue, purple, and magenta grapes, respectively. So spectra 1 was consistent with the expected structurally based blue and kingfisher reference data. So we found that HSI was a viable tool for assessing structural colorants, and this was the first time, to our knowledge, HSI had been used in examining not only Featherworks, but characterizing structural colorants within cultural heritage materials. So that was very exciting for us. Spectra 2 agreed with the structurally based purple feather coloring in the literature. But what really was interesting for us was Spectra 3 had a similar reflectance maximum peak to the same overall region area of that of the purple feathers, but we had some additional features present that led us to have some additional research questions. Specifically, the steep inflection around 600 nanometers led us to believe that an additional material, such as maybe a pigment, was present, and so that there was this combined pigment-based coloring and structurally based coloring. Whether we weren't sure if, as of yet, whether this was coming from a feather material itself, as I mentioned in the beginning of the talk, as feathers can have that combined system, or if it was present from some additional material the artist had added to the panels, such as a pigment of some kind that wasn't from the feather intrinsically. So that was a research question that I'll get into further later that we were really excited to explore. For the green feather areas, again, we turn to hyperspectral imaging. For this digital microscopy shown here, that highlights the sort of iridescent nature of these green feathers that we found throughout the panels. And so the reflectance from the HSI data indicated we had a reflectance maximum around 510 and 525 nanometers. And that combined with the overall visual cues and the shape of the feathers, barbules, and barbs indicate that the feathers were likely either mallard duck or peacock. So a really great benefit of these objects being at the Field Museum of Natural History is that we had access to the ornithology department there. So the ornithology department, in addition to having a team of wonderful scientists that have been super helpful throughout this process, facilitating discussions and use of equipment, they also have one of the largest collections of bird and reference samples in the world. So we were really lucky in that once we had identified a hypothesized list of species, we had access to this reference collection to further verify our hypotheses and really examine these reference samples with additional analytical techniques. So with this, we aim to get reference bird specimens for the blue, purple, and magenta and green coloring, hypothesized to be the common kingfisher for the blue, black-capped or purple feathers of kingfisher, and for the magenta coloring, we thought it could be the verticapped kingfisher and the green feathers were either the mallard duck or the peacock, as discussed previously. So our first step in assessing these reference samples, shown here is the common kingfisher, is we did a technique known as fiber optics reflectance spectroscopy to better look at the overall optical properties. And in particular, it allowed us to examine that angular base dependence that I mentioned previously. It's important to note also when comparing the FORS and the hyperspectral technique that these are very distinct techniques with different lighting conditions. So they cannot be compared directly on a one-to-one basis. However, we wanted to see if there could be conclusions drawn and the identification of the feather species from comparison of these data was possible, as often we don't have availability to every single instrumentation technique. Additionally, as I mentioned, HSI have been used to examine Featherworks previously, so we wanted to test it as a proof of concept and begin the process of developing best practices in the event that it was viable for examining Featherworks. And so we found for an agreement with literature that the maximum reflection peak was around 520 to 550 nanometers, and that as we go from larger and larger off-normal angles of incident illumination, the maximum intensity of that peak shift to shorter and shorter wavelengths, consistent with the angular response we had seen in the literature previously for these blue feathers. And then we found, when we compared that with the blue panel regions on the panel, the reflectance maximum was in the expected range of the common features. So we were fairly confident that this was an agreement and we were seeing the presence of the common kingfisher feathers in the panels. So to further verify this comparison and better demonstrate this proof of concept of using HSI, we compared modern reference samples with the panel directly, as shown here in this graph, and excellent agreement between these two areas further verified that this was an appropriate methodology for the work. And so we found that while, again, we can't compare FORS and HSI data on a one-to-one basis, we found that while relying on historical documentation of the material used, morphological comparisons, careful consideration of lighting and sample geometry, as well as our discussions with the experts in the ornithology department at the Field Museum, we were able to compare these techniques and use them for the identification process on the remainder of the colorants present on the panels. So then we completed the same comparison method of FORS and HSI data for the other types of feathers. So for the purple regions here, for the black-capped kingfisher reference samples, again, we see a shift in the maximum reflection value to shorter and shorter wavelengths as we go to larger and larger angles. And we find, once again, our hypothesis is correct that it was indeed the black-capped kingfisher based on the agreement between the maximum reflectance values between the FORS and HSI data. For the green feathers, in comparison of the FORS and HSI data, along with discussions with the ornithologists at the Field Museum, we identified the green colorants of feathers on the panels to be most likely mallard duck. Finally, in comparison of FORS data for the ruddy kingfisher and HSI of the panels, we see something different. So I mentioned previously that we weren't sure if an additional material was present for the pigment region of the magenta panels, and we wanted to better understand if this was coming from the feathers or some additional material. And with the FORS data, we got our answer pretty quickly because we see for the ruddy kingfisher that the maximum reflection is actually more into the UV in contrast to the panels, which has its maximum reflection on the shorter end of the visible. Additionally, we see a very distinct difference around 600 nanometers where there's an inflection point in the HSI that's not present within the ruddy kingfisher reference samples. So this led us to believe that an additional pigmentary element outside of the feather's natural biopigments was used in the panel's creation. So to further investigate the presence of a pigmentary element or painted components on the magenta feather regions, we turned to X-ray fluorescence. So when we mapped the region of interest here on the panels with the purple, magenta, and blue grapes, we found the presence of mercury on the panel, but only in the areas with magenta coloring. This combined with the HSI data confirmed the presence of vermilion. And in fact, upon closer examination, we can see the underlying base of the material here in the digital micrographs so a painted region of vermilion with purple barbules layered over top, that while at very zoomed in regions, we can see those two distinct colorants from a distance produce that overall magenta coloring or mixed coloring within our visual representation at the macro scale. So the reason for having this mix of materials, it could be as a desire to either mitigate the cost. Kingfishers are very small birds and they were very scarce at times. So that would be to minimize the use of resources. And also we've seen historically in documentation, the artisans and artists that crafted these works often used foils or underlying backing materials to alter the visual appearance. So while they didn't have access to the same analytical technology we do today, they had a really great understanding of their materials they were using from a qualitative perspective, and they were able to manipulate them to create these desired optical effects. I won't get into the other pigments and materials we identified too much because I want to get into our synchrotron-based work on the feathers, but I want to highlight some of our other findings briefly here. So with that same multimodal approach using XRF and HSI, we were able to identify other pigments present on the panels in the painted regions. So for the pink, we found that it was an insect-based pink, either most likely a lake or a cochineal, and the white on the panels was most likely lead white. We again found vermilion present on the panels, and we found two distinct different types of green, a copper-based green, most likely malachite, and then another green that was in presence of a mixture of indigo and some type of organic yellow. These panels, I haven't had time to showcase all of what they had because of the scale and the details, but they had so many different interesting elements, including numerous types of gilding and adhesives used to adhere the Featherworks. So I wanted to just highlight these very briefly. There was 3 different types of gilding on the panel. So the top row here is sort of just an image of these different regions, and the bottom are different micrographs really highlighting the details of some of these gilding features and adhesives. So 3 different gilding types were found as detected by XRF. The first was a zinc copper alloy. The second was zinc copper alloy with some gold adornment on top. And finally, the last type of gilding was a silver tin-based alloy. Additionally, we use Fourier transform infrared spectroscopy to identify adhesives on the panel, and we found that it was in excellent agreement with fish-based glue or isinglass, which is well given to be known during that time period. So we had successfully identified the species of birds and characterized the optical and morphological properties of those feather-based materials, along with examining the overall materiality of the kingfisher works. But we really wanted to see if we could push this even further and better understand these optical materials and really develop and correlate structure property relationships between the observed coloring and the overall structure morphology. So we had investigated these all using the techniques previously discussed of SEM, HSI4s, but we wanted to see what other techniques were available to us and how we could really even push this further. And so to that end, to better understand the material properties, we turned to 2 techniques specifically, optical goniometry and synchrotron-based ultra small angle X-ray scattering. So in collaboration with the Chazot lab at Northwestern, we used their optical scatterometer. So optical scatterometry or optical goniometry is a reflectance spectrum measurement device. And what's really unique about it is it measures the reflectance spectra of a sample over a range of angular values. So I discussed previously with the FORS data, we were able to measure those within 15-degree increments. But with how the optical goniometry works, you have your sample in the center, and then the light source is at a fixed angle that you choose as the investigator. And then the detector actually moves in a 180-degree arc pathway. So you're actually able to get reflectance spectrum measurements for samples with 1 degree or even 0.1 degree resolution in the case of some systems. So it really allows you to capture that angular variability, which is particularly important for the structurally based materials we've been seeing. So we performed these measurements on reference kingfisher samples, and what this chart here represents visually is that we converted the reflectance spectrum measurements to RGB color space, and then we plotted them at the corresponding angle at which they were recorded. So the variation of colors and hues within the blue region that you see along this arc path is actually very similar to what you would see is if you tilted a feather sample and were able to see that visualization or change in coloring from the feather itself. So we selected the range because it was most representative of the sample, and we really wanted to highlight and visualize this dynamic range because it's particularly important in Chongqing and other Featherworks. I mentioned how well that the people fabricating these works knew their materials. Oftentimes in Chongqing, the headdresses or jewelry, so in some cases, were even on springs or were very dynamic and designed to move with the light and play on these interactive angular varying properties. So we really wanted to see how well we could represent the angular changes to really show and highlight the dynamic nature of these materials and also just illustrate their overall materiality in the best way possible. So in investigating these materials, we also began a collaboration with Argonne National Laboratory to do ultra small angle X-ray scattering, or USAXS or SAXS. So we were specifically interested in USAXS for structural color as for a variety of reasons. So first and foremost, the scattering vector Q is inversely related to the size and morphology of the sample. So similar to what we can get with SEM, we can get information on the size of features and the spacing or the periodicity of them. But in contrast with SEM, SAXS can go even further in telling you overall information about the order of the system. So you can get information on the overall order of features and really highlight some unique differences. Furthermore, there's minimal to no sample prep needed for reference samples at the particular beamline that we used. And finally, no sampling is needed in contrast with the SEM methods discussed previously. And for working with cultural heritage samples, the idea that we could perform these techniques without the need of sampling and it would be non-invasive was really exciting for us. So the specific setup that we used at Argonne contained optical cameras to allow us to visualize where we were in terms of the color of the feathers with the measurements that we were taking. So we could also use burn paper as a spot size to understand the more precise beam location with the camera. The optical cameras that we do have in the setup, they do have a bit of parallax, but luckily for us, the instrumentation also has X-ray radiography imaging. So if the structure can be seen with X-rays, then we can have resolution down to 10 or 20 microns. That way we can really correlate where we are visually within the sample with the overall structural data we get from the USAXS measurements. So on the right here is a representative example of the USAXS data I'll be discussing throughout the rest of this talk, where we have the intensity plotted as a function of the Q. And so here, the primary features I want to note is that there's this diagonal line that's coming from the overall background of the material and the diffraction peaks here that are associated from the ordering features in the material system analyzed, and that they are inversely related to the size of the material. So a larger Q corresponds to smaller materials and vice versa. I will note the energy that we used here was 21 kilo electron volts and the beam size was 1 millimeter squared. So our initial experiments, we aim to assess the feasibility of USAXS on our reference sample. So the figure here represents data collected across a common kingfisher feather from the blue to brown gray region as indicated by the arrow. So we wanted to see if we could successfully capture the structural features and assess them first and foremost, and then also if we could correlate that to visual changes we see within the feather. So we see with the graph here with the intensity plot as function of Q, with this inset here just being more highlighted of this particular region for clarity, that as we go from the blue to the brown gray region, we see the disappearance of this primary diffraction peak. So this highlights that as we're seeing the disappearance of the peak, we can successfully identify the blue structural color and attribute that primary diffraction peak to the blue structural color in our system. And also, since we have this at the same location within the Q space, that means we're able to quantify the overall spacing of the features in our system using USAXS. So that was very exciting for us that this does prove feasible as a method to examine structural color in feathers. Our next step in assessing the feasibility is we needed to assess the system's overall anisotropy. So the anisotropy in the system in relation to the blue color is, I mentioned that these are very periodic and ordered systems. So we wanted to make sure that any changes in either the feather geometry, if they were rotated or off axis, that wouldn't impact the measurements we are seeing, because we wouldn't want to attribute any setup on variation or orientations to our measurements versus actually identifying what's important for the intrinsic material itself. We tested many different orientations, but I only showed 4 here just for simplicity and better visualizing of the variation. And we found that there was no systematic changes for the different orientations, meaning that so the results and the slight differences here are within expected natural variation. So that means that we do, in fact, not necessarily care about the feather geometry, which for reference samples, this is maybe not as critical because we can place or precise orientation of the feathers. But once we move to our cultural heritage samples eventually, because they're already adhered to a surface and we can't manipulate the object, that was really exciting to us that this is still potentially a viable technique for direct measurements on cultural heritage works. I will point out here that the reason that we might have had such success and we didn't have to factor in the overall orientation and geometry is we were using a slightly larger beam size, and also that, as I mentioned, the Kingfisher system is somewhat disordered and has that nano sponge system. So in other structural colorants, the orientation could be a greater factor and is something to always assess before you move forward with your measurements. So as we often know, getting to the beam line can be a challenge in itself, and getting beam time and also just ensuring that the data is collected properly is a challenge, but oftentimes that's just the first step. In my experience, getting the data and collecting it is time intensive, but actually processing all of the data you get from a beam line and knowing what to do with it, how it fits into the puzzle, and modeling all of that data to be useful for understanding the materiality of your objects is really what is the most time-intensive process. So I wanted to go through a little bit how we model our USAXS data, specifically what software we used, and the key model metrics we looked at when assessing the data we collected. So the USAXS data, we used a specific software called Igor Pro. And in fitting the USAXS data for our system, we used 2 different models. One was a unified level for background, so this matches the overall shape of this diagonal feature. And the second, which I'll get more into because it has more metrics that are useful to us in assessing the color of our systems, are primary diffraction peaks. So with the primary diffraction peaks, as highlighted here, there's some key model metrics. First is the Q location. So where is that peak occurring? I mentioned the location of that spacing can be related to the size of the materials. So from the Q information, we also can get D spacing, or the periodicity of our samples. We can look at the full width half max of the peaks, and that relates to the color range or the spectrum width of the colors that we observed in order of the features. We can look at the intensity of the peaks, and this tells us information on the overall color saturation or brightness. And then finally, we can also look at the overall number of peaks. So we see for this representative example here, we have not only a primary diffraction peak here, but also secondary and tertiary peaks. So the number of peaks can tell you the relative amount of order in the system, with the number of peaks increasing means the system is more and more highly ordered. So this here is a representative blue example for the common kingfisher. And we found the D-spacing to be around 205 nanometers. And this was great because that was in line with our previous scanning electron microscopy results and the quantitative assessment we had done on those samples with 4A analysis, and also was in agreement with the current literature related to common kingfisher measurements. So we then examined the reference samples from the 3 hypothesized kingfisher species: the common kingfisher, the black-capped kingfisher, ruddy kingfisher, as plotted along the same graph here. And we see, as we go from the blue to purple to magenta coloring, the primary feature is at larger and larger Qs, and therefore associated with smaller and smaller nanostructure periodicity and features. So with the USAXS Synchrotron methods, we can not only successfully identify the structural colorants in these features, but we can also distinguish between these three species using a combination of techniques. And that was exciting to us that we can now have this multimodal approach where these techniques can be combined synergistically to not only distinguish and identify the species, but also aid in better understanding materiality and their properties. So modeling for each of these systems. So this data is still in the early stages of processing and quantitative analysis, but we can already start to draw some really interesting conclusions from the initial modeling. I know a lot of the screenshots can't be visualized here, but I've highlighted the most key metrics of the D-spacing in the text at the bottom of each graph. So we found that for the common kingfisher blue samples, the D-spacing was around 205 nanometers. For the black-capped kingfisher, 170 nanometers, and for the ruddy kingfisher, 155 nanometers. Additionally, a key distinction to note is the differences in the secondary and tertiary peaks within the systems. So for the blue and purple colorants, we still see that the secondary, tertiary peaks are present. However, when we go to the magenta colored feathers or ruddy kingfishers, we see that those features are really no longer present. There is one secondary peak we can identify, but it's much weaker in intensity. So this indicates that the overall nanostructure system is more highly ordered for the blue and purple coloring, at least over larger length cells. So we're excited to work up this data further to better understand the quantitative metrics and relate those to the spectroscopic data that we collected and I discussed previously. Furthermore, I want to highlight that the results of this distinguishing between the purple and the magenta color really supports the analysis that I discussed previously, that in fact the magenta coloring here is not from one individual feather component, but in fact a pigment, in this case vermilion, and combined with purple feathers. So structural colors are often regarded for their stability and something that they're very resistant to UV fading. They're more robust than dyes and pigments, but this isn't always investigated in a very systematic fashion. One example of structural colorants in ceramics that I wanted to highlight are the silicon oxycarbide polymer-derived ceramics that were published previously, where a non-iridescent structural color was produced, and it was found to demonstrate thermal stability up to 1,000 degrees Celsius for 100 hours. So in this case, there was a very systematic test of the structural color stability in these ceramic materials. However, feathers, as mentioned in the beginning of the talk, often contain pigmentary colors in those mixed systems, and even in solely structural systems, the ordered features often stem from melanin in the melanosomes or other biopigments. So those, because they're composed of pigments, that structural color stability might still be prone to some of those same degradation mechanisms. And even if we don't see dramatic differences in coloring, that doesn't necessarily mean no degradation is occurring. So our next step in this project is we really want to better understand the stability and what we can do to characterize this and connect it with changes in observed coloration and maybe alterations in materials. So this is one of our project's next goals that I wanted to highlight and is in the preliminary stages of being investigated. So as I mentioned, one of our next steps was to investigate the degradation in structural color. Shown here are some preliminary results for 2 unaged and 2 UV-age samples for the blue common kingfisher. The UV-age time was pretty short, around 4,500 minutes. So we didn't see any distinction between the unaged and UV-aged short time samples. This isn't entirely surprising, as we know that structural colors are more light fast and less resistant to UV-based degradation. However, we still believe SAXS is a great option for investigating these changes, as it can capture changes on the structure beyond just overall visible color changes. And we think some of the key metrics that we have in our model, such as full width half max, the order of those peaks could change, the width or intensity could change, and so we could start to see and visualize those changes to better understand degradation mechanisms and elucidate those phenomena. Additionally, our next steps in this project are going to be moving on reference samples and working on cultural heritage objects directly. So we've already identified some objects well suited to USAXS measurements at the Field Museum of Natural History, and our selection of suitable kingfisher featherworks based on the requirements of the bead and lime setup. We've completed some preliminary XRF analysis to identify the backing materials, and we're in the process of preparing mock-ups for parameter optimization to understand how those backing materials may impact the energy and beam size we need to use in those measurements. I wanted to note in this case, we'll probably be using small angle X-ray scattering instead of USAXS because of the presence of those backing materials as well. So it's been a really pleasure today talking to you about some of our recent work investigating feather-based materials and the multi-analytical approaches we've been able to apply to these materials, in particular the synchrotron-based methodologies. And so we've successfully analyzed feather objects with both pigments and structurally based coloration throughout this project, comparing the screen panels, reference samples, and bird specimens to not only identify the species, but also understand their overall materiality. We've been able to develop methodologies to better examine featherworks and structural colorants using hyperspectral methods for the first time. And we also were able to assess the nanostructured features capable of producing these colorings using synchrotrons, USAXS, and SAXS methods. So our next steps are to optimize these for direct measurements on cultural heritage works and also look at degradation measurements in the goal of part of a pilot project to create feather and structural color databases. So within all fields, it's always a highly interdisciplinary work, but I know heritage science especially relies on a very large team and collaborations with different institutions and expertise. So I wanted to highlight and thank my colleagues, not only at the Art Institute and the Northwestern Center for Scientific Studies of the Arts, the beamline scientists I've been working with Argonne National Lab, those at the Field Museum of History, and also our funding sources here for making this work possible. And I'm happy to take any questions now. Thank you so much.
Thank you so much, Madeline. That was such an excellent talk. And I love seeing the photos of the feather reference collection. It looks so delightful. We're able to take questions. We have about 13 minutes. So if you have any questions in the audience, please use the Q&A section. I know that already we have one. While we're waiting for more, I wanted to ask a little bit more about the feathers and the way that they degrade. So could you talk a little bit about the composition of these nanosponges and what are the factors that might cause them to alter in structure and then affect the structural color?
Yeah, that's a great question. So there's sort of two for structural colorants and feathers in particular. There's kind of two classes. So the iridescent colors are formed often by melanosomes. So those are melanin granules that have other chemicals present, but they do have melanin within them, which is a biopigment. And then for the kingfishers, there might be some melanin present in like a basal layer, but they're actually composed of a keratin air matrix. So most likely the degradation pathways would be due to like breakdown in the morphological features versus like chemical photoaging. So the same degradation that you might have with other keratin or materials that we see would probably still impact here. So that's why I think USAXS and SEM are particularly important because we might still see the color is maintained, but there could be some morphology changes of that matrix. It might be becoming more brittle, and maybe it's that they're more robust to light-based in damages, but maybe temperature or mechanical abrasion, that's going to be where some of the issues occur. So I'm excited to look into that further.
Yeah, that's cool. And did you notice any areas where you might potentially see degradation in the featherworks themselves, or were they very well preserved overall in the objects that you were looking at?
We did see some color variation, and some of that we weren't entirely sure it was in part from natural variation. So part of our next upcoming measurements are also going to be doing collection of reference samples that are historical and modern to really get an idea of the range of natural variation can occur. Because we even see with different chung tree objects that those in the highest craft are very uniform in color, but some that are maybe still very well executed have a little more variability. So it seems like for the most prized works, they're very consistent in the coloring because they were only used in certain regions of the feathers. So that natural variation also plays a role.
Yeah, that sounds so cool. Yeah, it makes complete sense also. Okay, so we have one question from the audience right now. In your scatterometry method, in addition to angle measurements, were you working at a range of distances?
For our setup, the light position is fixed, so we can vary with the lens that's on it, the overall beam size. We had it be, I believe, around a 0.5 millimeter squared, just because that allowed us to get the signal to noise we needed for the system. But you can go to smaller beam sizes. Um, in the particular configuration that we had in collaboration with the Chazot Lab, their instrumentation, to my knowledge, had pretty much a fixed distance, or at least what we needed for the signal to noise. But that could be something that you could vary. And I'm hoping to maybe create a customized for setup that allows for more angles than the 15-degree increments where the distance would be able to be altered.
Yeah, okay. That makes sense. Also, when you're dealing with three-dimensional objects, that's something that would be very important then for focusing.
Yes.
Yeah.
Yeah, you have to be very precise in when you do the angle, you want to be along the barb of the feathers. You don't want to be going like perpendicular to them. So that's something that's also important because that can lead to differences in the measurements. So when we're developing the best practices for these measurements, which a lot of that's already out in the literature as well, Ellen Perlstein in particular at UCLA.
Oh, yeah.
I've talked with her. She's done some really great Featherworks and discussing the impact on cultural heritage works and feather geometry. But that's also something that's very important to keep in mind.
Okay, maybe another question from the perspective of, and I know you touched on this about structural color in ceramics, and thinking about this as structural colorants within an like amorphous silicate glass. I'm just thinking, how do you think the light reflection from the glaze itself, from the glass component, might affect the structural color, I guess, assessments?
No, that's I'm not as familiar with the glass, but I know it acts as like a photonic glass in
Okay, yeah.
principle. And so that at least for some of the materials, but we have found that maybe different like glazes or varnishes for paintings and top, that because of the changes in the refractive index of the materials, that's also impacting the overall structural features. So that's very critical. The specific example I mentioned, those were polymer derived ceramics. And I think that they were doing a like multi-step layer process to fabricate those, but I need to look into that a little more a little further because I'm not as familiar with ceramics. It's not quite my expertise
Yeah.
in structural colorants, but I know there are a lot of structural colorants in the iridescence within those ceramic materials.
Yeah, no, and definitely for like stained glass windows or any kind of colored glass, it wouldn't even need to be like a ceramic glaze. I could see this being really useful, very important way to track how this is angular dependent. So yeah, I'm excited. Are you hoping to work a little on ceramics in the collection?
Yeah, I think ultimately, I think I've been focusing on Featherworks for right now because once I started on Featherworks, I just got then more people, started and I got just so excited seeing the diversity of works, the combined pigment and structural color. But it would be like a goal or dream of mine to expand to more types of structural colorants. They're including ceramics and also even other natural materials such as butterfly wings or beetle shells, which also exhibit these really unique properties.
Definitely. I've been doing some work on Indian manuscripts and they have little cutout of beetle wings in some
Yes.
cases, and they're really gorgeous. They're so tiny though. So being
Yeah.
able to resolve like that type of like dimension, I think it would be super cool. I'm excited to see what you do. Okay, we have one more question. How does this data relate to the culture? This is a great question. Are there any implications on how or where these feathers were acquired for producing the artifact? Is there a specific reason why these feathers or colors were used? And did they carry any meaning for the culture?
Yes, so I can speak to that. They definitely did have a meaning to the culture. Depending, it is very interesting too. Something we've been working and talking with some art historians as well is how this might have varied across different cultures. So for the Changchui objects, what was very interesting in contrast to the Peruvian like tunics and textiles I mentioned at the beginning is they use the individual pieces of the feathers versus the feathers as a whole. So that was an interesting distinction I noticed that In the Chang works, it's more the individual pieces of feathers, but we know that this color was very symbolic to them, and also that it's mentioned not only, we did a deep literature search using and with some computer scientists to find reference of kingfisher blue, and that's a common color that's used not just in fabricating works, but also within poetry. So it has a lot of meaning, the kingfisher, as well as the association with that blue, kind of like, I feel like in English, we have like the robin eggs blue, like it clearly is like a very distinct color. And also these birds were often, they used individual pieces from the birds themselves. So they would acquire the birds and then use the ones that had the appropriate coloring. This did lead to, at some point, almost extinction of some of these species.
Oh, no.
At some point during the height of their popularity, and they had to go to different regions within Southeast Asia, which seems to be okay now, but it was at the height of their popularity a bit of an issue because they're very small birds, and they not all of the region of the birds has this bright coloring. So that was the material scarcity, especially for some of these large scale works. You would need quite a lot of feathers to create something at this level.
Wow. Are there any, as far as you know, artists that work on synthetic feathers? Is there a way to, I guess, have similar optical effects?
So it had, there are cases where conservators, when they do retouching, or artists today will dye feathers in terms of capturing a structural color. And we've actually seen in cultural works, especially within indigenous communities, Native Americans, they dyed eagle feathers sometimes in their regalia. But in terms of producing a structural color on feathers, I'm not quite sure yet, but there are starting to be commercialized structural color paints. They're used on some cars to kind of get that pearlescent effect. So biomimetic materials mimicking this natural properties are a really highly active area within material science. And I think that also, for me personally, really contributes to my interest in the field, because seeing how these materials degrade or how their optical properties can be manipulated, I think can also not only inform our understanding of these cultural heritage works, but also help inform material scientists or physicists or chemists who are developing these next generation of bio-inspired materials.
Yeah, no, that's so relevant then to modern applications. There's one question, and I think that's actually for me. The link for the conference, I'll send that now. And the link for joining AAC. Okay, so the link that is currently on the chat is for graduate students and doctoral students that are not currently members of ACerS that would like to become members, because as a division, we're able to cover membership for a year. We are not able to cover membership for other people if they're not graduate students. That's the only thing. So if you are interested in becoming a member of AACS, it would be through the typical ACerS membership process. And once you become an ACerS member, you can choose the AACS division as your host division, as your the one that you're assigned to. But yeah, I'll send out actually, Vicki, do you have the link to MS&T? I'm not sure if I have it directly on me now, and we are reaching the end of
Yes, I will
webinar.
pull that up and share that in the chat here in just a moment.
Okay, thank you. But yeah, anyway, there will be a recording. Well, there potentially will be a recording available. I believe that Madeline needs to review it first. But we're hoping to be able to upload it onto YouTube. So just wait and see. We need to make sure we get the right permissions from the museums. And yeah, I think while we're waiting for Vicki to put that link for the MS&T symposium, I will just start thanking Madeline for such a fantastic presentation and insight into the details of the nanostructure and the color dependence of feathers. It was great to have you here, and we're honored to have you as the second speaker of the webinar. There is going to be another webinar in a month's time, and our speaker is going to be looking at ancient glass. She's currently a postdoc at the Met. So I'm really excited to welcome Ellen Purdy to give a talk next month. But for now, let's just thank Madeline one more time, and I hope to see you all next month.
Yeah, thank you so much to everyone for being here. It's very exciting to share my newly found interest in Featherworks these past few years. It's a fascinating material, and I love to talk about with anyone when I can get the chance to. So,
thank you. Okay, let's pause the recording or stop the recording. Okay.